Hello friends, welcome back to another video. In this session, I am going to explain a question regarding the process of polyadenylation or tailing during transcription. Actually, this question is asked in 2015 NET Life Science examination and it is an analytical question given in part C. This question is also very important for NEET aspirants because this process of tailing is also a part of your syllabus. Let's directly move to the question. And the question is, the 3' end of most eukaryotic mRNAs is defined by the addition of a poly-A tail processing reaction called polyadenylation. The addition of poly-A tail is carried out by the enzyme poly-A polymerase. Given below are few statements about this process. And this question is regarding tailing during the process of transcription. And before coming to the options, first of all, we should have some idea about the process of transcription and also some terms like DATP and ATP. First, we can check what is the difference between DATP and ATP. You know, DATP is Deoxy Adenosine triphosphate and ATP is adenosine triphosphate. First, we can check what is this Deoxy Adenosine triphosphate. Actually, this Deoxy Adenosine triphosphate is having two portions. That is Deoxy Adenosine part and it is having a triphosphate. Now we can check what is this Deoxy Adenosine. You know, Deoxy Adenosine is an example for nucleoside. You know, nucleoside is having two components. They are pentose sugar. First component is a pentose sugar and second is a nitrogenous base. And we can consider this pentose sugar and nitrogenous base in this deoxy adenosine. So this is a pentose sugar and also you know this is deoxy adenosine. Deoxy adenosine will be having a pentose sugar. That pentose sugar will be deoxyribose. Since it is a deoxyribose, in the second carbon atom, you know this is first, second and third, fourth and fifth carbon atom. This is a sugar, pentose sugar. This deoxyribose lack hydroxyl group in the second carbon atom. Instead, it will be having only hydrogen atoms in the second carbon atom. There is a lack of hydroxyl group in the second carbon atom, so that we can consider this sugar as deoxyribose. You know, the nitrogenous base in this deoxy adenosine will be adenine. So this adenine plus deoxyribose together constitute deoxy adenosine. And this nitrogenous base adenine is linked to the first carbon atom. In the first carbon atom, you can see nitrogenous base, that is adenine. This adenine, so you know this is nitrogenous base and this part is a deoxyribose sugar. This deoxyribose plus nitrogen base adenine together constitute deoxy adenosine. Deoxy adenosine. We are discussing about deoxy adenosine triphosphate. So the first part is over, that is deoxy adenosine, and now we have a triphosphate. You know, the triphosphate means there will be 3 phosphoric acid groups. That phosphoric acid group will be linked to the fifth carbon atom. You know, this is the fifth carbon atom. This fifth carbon atom will be having 3 phosphoric acid group. 3 phosphate groups are there, so that this 3 phosphate, that is called triphosphate, along with this deoxy adenosine together constitute deoxy adenosine triphosphate. This deoxy adenosine triphosphate we can write like DATP. This is the deoxy adenosine triphosphate. How to get a DAMP? This is deoxy adenosine monophosphate. We can easily get a DAMP from the DATP. You know, DATP means it is deoxy adenosine triphosphate and DAMP means it is deoxy adenosine monophosphate. So we have to remove 2 phosphate group. If you observe the structure of this, we have seen 3 phosphate groups in this part. From these 3 phosphate group, the 2 terminal phosphate groups can be removed to form a DAMP. Once 2 phosphate groups are removed from this, we will be getting DAMP. Here we will be having only 1 phosphate because we have removed 2 phosphate. This is called DAMP. Remember, this deoxy adenosine monophosphate is also called deoxy adenylic acid. We are also having another name for this compound, that is deoxy adenylic acid. Deoxy adenylic acid. Remember, this deoxy adenylic acid is an example for nucleotide. Nucleotide means it is having a pentose sugar, a nitrogenous base, and there will be a phosphate group. You can see there is a pentose sugar. This is a pentose sugar, and also there is a nitrogenous base and phosphate group. This nucleotide, remember this deoxyadenylic acid. This deoxyadenylic acid is a monomer of DNA. That is our point. It is not a monomer of RNA, because RNA is having ribose sugar. Now we can come back to the ATP. If I explain what is DATP, and from this DATP we can get DAMP by the removal of 2-phosphate. In the same way, you can also easily say that what is ATP and what is AMP. And if you take ATP, ATP is also called adenosine triphosphate. Actually, this is made up of adenosine plus triphosphate. First, we can draw the structure of adenosine. Adenosine means it is having a pentose sugar. As I explained earlier, adenosine is also an example for nucleoside. Adenosine is also an example for nucleoside. Nucleoside is having a pentose sugar and also a nitrogenous base. But adenosine will be having a pentose sugar. That pentose sugar will be ribose sugar. But in deoxyadenosine, the pentose sugar was deoxyribose. But now we are explaining adenosine. In adenosine, the pentose sugar will be a ribose sugar. This will be a ribose sugar. And also the nitrogen base will be adenine. The nitrogen base is adenine in this adenosine. If we consider the structure of deoxyadenosine, this deoxyadenosine is having a pentose sugar. This is a pentose sugar, and also it is a ribose sugar. Since it is a ribose sugar, this is first, second, third, fourth, fifth carbon atoms. In the second carbon atom, there will be a hydroxyl group, so that we can consider this as a ribose sugar. I think you remember in deoxyadenosine, we have seen a deoxyribose, so that here we were having only hydrogen atom. Hydroxyl group was absent. Due to the lack of this hydroxyl group, we call this ribose as deoxyribose. But here we are having a hydroxyl group, so we can call this pentose sugar as ribose sugar. Then the next part is adenine. To the first carbon atom, an adenine is attached. This is a nitrogenous base. This nitrogenous base and pentose sugar together constitute adenosine. Adenosine is an example for nucleoside. And we have to add triphosphate. So to form a triphosphate, we can just add 3-phosphate group to the 5' end. Now you can see there is an adenosine, and also there is 3-phosphate. This is called adenosine triphosphate. This adenosine triphosphate is nothing but ATP. How can we get AMP from this ATP? Adenosine triphosphate is having 3-phosphate group, triphosphate. AMP is a monophosphate. So we have to remove 2-phosphate group. These 2 terminal phosphate will be cleaved. Means pyrophosphate will be removed, and we can easily get AMP. So AMP is derived from ATP. If you consider the structure of AMP, it will be having a nitrogenous base, that is adenine, 1-phosphate at the 5' end. So all these 3 together constitute adenosine monophosphate. As you know, this adenosine monophosphate is also an example for nucleotide. AMP is also called adenylic acid. And this nucleotide is a monomer of RNA, not DNA. In RNA, there will be a ribose sugar, because here there is a hydroxyl group that I have explained here. In short, we can conclude both DNA and RNA are made up of nucleotides. And the nucleotide present in DNA, that is called DAMP, deoxy adenosine monophosphate, and the nucleotide present in RNA is called AMP. This is a basic things, and also it is very, very important for answering this question. Also, you should remember, this DAMP is derived from DATP. You know, DATP means Deoxy Adenosine triphosphate, that donates DAMP by the removal of two phosphate groups. And this AMP is derived from ATP, that is adenosine triphosphate, by the removal of two phosphate groups. To answer the given question, you should also have some basic idea about transcription. You know what is transcription? Transcription is the formation of RNA from DNA. Actually, transcription process occurs during the expression of a gene. Let us consider this is a DNA strand. Its polarity is 3' to 5'. Using this DNA strand, we can synthesize an RNA molecule. The polarity will be 5' to 3'. Formation of RNA from DNA, this process is called transcription. You know what is the difference between transcription and replication? DNA replication is nothing but if you are having a DNA strand and we are going to synthesize another DNA strand using this strand as a template. So synthesis of DNA from DNA strand, this process is said to be replication, DNA replication. And synthesis of RNA from DNA, this is called transcription. As you know, DNA replication requires an enzyme, that is DNA dependent Rna Polymerase. In the same way, formation of RNA requires another enzyme. This enzyme required for transcription is called DNA dependent RNA Polymerase. Remember, RNA polymerase can synthesize RNA. That is why we are getting an RNA here. And also it requires a DNA strand as a template. You know, this is a DNA template strand. This process of formation of RNA depends on a DNA template. That's why we are saying that it is DNA dependent RNA polymerase. Now let's consider this DNA strand is going to be transcribed. Means this DNA is acting as a template strand to synthesize a new RNA strand. So this process is requiring an important enzyme, that is called DNA dependent RNA polymerase. There are different types of DNA dependent RNA polymerases. DNA dependent RNA polymerase 1, polymerase 2, polymerase 3. Suppose we are having a DNA dependent RNA polymerase 2. During this process of transcription, we will be getting an RNA here. If this is DNA dependent RNA polymerase 2, this will be forming an hnRNA. The product is said to be hnRNA. hnRNA is nothing but heterogeneous nuclear RNA. Its polarity will be 5' to 3' because the template strand is having 3' to 5'. And this hnRNA is actually the precursor of mRNA. You all know about mRNA. mRNA acts as a template for the synthesis of protein. During translation process, mRNA is acting as a template. Anyway, to get this mRNA from hnRNA, 3 different processes are required. The first process is capping, the second is tailing, and the third is splicing. I'm not going to explain all these processes. Actually, our question is regarding tailing, so we can just discuss tailing in this part. We can explain the transcription process in another video. The given question is regarding the process of tailing. Actually, what is process of tailing? Tailing is nothing but addition of a number of adenylate acid residues to the 3' end. You know, this is a 3' end. To the 3' end, we have to add a number of adenine residues. All these are adenylate residues. During the process of tailing, nearly 200 to 300 adenylate residues are added to the 3' end of hnRNA. And this process of addition of adenylate residues is called tailing. Actually, what is this adenylate residue? This adenylate residue is nothing but AMP. I have just explained the difference between AMP and ATP. I think you remember how can we get AMP? AMP is derived from ATP. You know the structure of AMP and ATP. I just explained. This AMP will be added to the 3' end of hnRNA to form a poly-A tail. This tail is said to be poly-A tail. Very important point here, the adenylate residues represent AMP and this AMP is derived from ATP. finally we are going to get a very long poly A tail. This process of tailing is catalyzed by another enzyme. This enzyme is said to be poly A polymerase. This enzyme is known as poly A polymerase. There is a very important difference between DNA dependent RNA polymerase and poly A polymerase. That is also very important. As you know, DNA dependent RNA polymerase synthesizes a new strand of RNA depending on a template DNA strand. But here you can see this poly A polymerase. This enzyme is also producing a number of adenylic acid. This is also synthesizing an RNA strand here. Poly A tail is nothing but an RNA strand. There is a similarity. You can see DNA dependent RNA polymerase synthesizes RNA and poly A polymerase is also synthesizing an RNA. But what is the main important difference? The main difference is this DNA dependent RNA polymerase requires a template strand to synthesize RNA molecules. But this poly A polymerase does not require a template. Means this process occurs in a template independent manner. So you can say that the process of tailing, we are discussing about tailing. Tailing is a template independent process. So two things I told. During tailing, AMP is used and this AMP is derived from ATP. And also the process is catalyzed by an enzyme poly A polymerase. And this poly A polymerase does not require a template strand. So we can say that the tailing process is a template independent process. If you know these much things, you can easily answer that question. We can come back to the question. 3' end of most eukaryotic mRNAs is defined by the addition of a poly A tail, a processing reaction called polyadenylation. The addition of poly A tail is carried out by an enzyme poly A polymerase. Given below are few statements about this process. Now we have to check these statements A, B, C, D, E, F. And we have to identify the correct combinations. The first statement, poly A polymerase is a template independent enzyme. You can easily say that the first statement is correct because poly A polymerase is producing RNA without using a template strand that I just explained. The second statement, poly A polymerase catalyzes the addition of AMP from DATP. I think you remember AMP is derived from ATP, not DATP. You know DATP will be providing DAMP. So you can say that the statement B is wrong. Poly A polymerase is an RNA template dependent enzyme. This enzyme doesn't depend on DNA or RNA, means it is a template independent. So this statement is also wrong. Next statement, poly A polymerase catalyzes the addition of ADP from ATP. There is a small mistake. This is not ADP. Actually, poly A polymerase add AMP, adenosine monophosphate. ADP means adenosine diphosphate. So that this option is also wrong. Next statement, poly A polymerase catalyzes the addition of AMP from ATP to the 3' end of mRNA. I think you remember this statement is correct. AMP is linked to the 3' end. So the statement E is correct. Next statement, poly A polymerase catalyzes the addition of AMP from DADP to the 3' end of mRNA. Here the first part is correct. It is adding AMP, but it is not from DADP. Instead, AMP is derived from ATP. So the last statement is also wrong. Then which will be the correct option? A and E are correct. The correct option is 3. I hope you understood. If you like this video, please subscribe and enable the bell icon to get my upcoming videos. Okay, we can meet in another video with another topic.